Hello, my name is Gabriel Dunn. Today's date is August 9th, 2018. In this week's video lab in microbiology, we're going to be investigating basically how the environmental factors will affect how a microbe can grow. For example, in the exercise 1 and 2, we're going to be talking about how an acidic environment, can inhibit or allow or even promote the growth of certain microbes, as well as looking at isotonic and hypertonic solutions of sodium chloride broth. And basically we're going to inoculate it with the S. cerevisiae and S. epidermidis that we've been culturing, and we're going to see if these two microbes can grow in these two different environments. Okay, so I'm going to go ahead and demonstrate. Okay, so we're going to be using aseptic techniques to do a pure culture of this, so we know that it's that microbe that we're testing. Okay, so the first thing I'm going to do is go ahead and I'm going to adjust the pH of one of these broths. I'm going to take the one that needs to be a pH of 5. Okay. And so for that, I'm going to go ahead and use the hydrochloric acid, just 1 drop of that. So I'm going to go ahead and adjust that. Gonna flame the lip. And add precisely 1 drop. Okay, so the pH is now adjusted to a pH of 5, so we're going to close the lid on that. And I'm going to do the same with the second vial. Okay, so I'm going to flame the lip with that. And get the hydrochloric acid, precisely 1 drop. The pH of the broth is actually pretty much neutral. It's 7, the pH is 7. So we don't actually have to do anything to that tube because it's neutral. Okay, so now I'm going to add 2 drops of each of the microbes to each one of the tubes and then incubate those for 24 to 48 hours. Also using still techniques, especially on the cultures here, the active cultures. We don't want to get those contaminated, because then we won't know what we're testing for. We're testing with. Okay, so we're going to go ahead and take that and match the microbe. This one I grabbed is the S. epidermidis. So we're going to match it with that tube, of course. And we're going to flame the lip. I'll return the caps. Okay, so that one's done. And do the same thing with the cerevisiae. pH of 5. Two drops. Flame the lid before returning it. Okay, so pretty much the same thing's gonna happen with the sodium chloride tubes. As you see, I've labeled them here. I have the sodium chloride for each species and each concentration of solute. So let me go ahead and do one of these really quick. I mean, it's not much. I'm just gonna put 2 drops of the microbes in each tube. So right here I have the 1%. This is like pretty much like isotonic. So 1% of each one. So we're gonna do that real quick. Sterile techniques, of course. Using the same specimens. This one's the first one I'm doing is S. epidermidis. 2 drops. 1 or 2. Okay, gotta flame the lip. Return the cap. And also for these, we're going to go ahead and incubate them for 24 to 48 hours. We're going to check this for turbidity. Going to see if there's any growth. And if there's any growth in that time frame, then that indicates a positive test result. If there's no turbidity, no growth, then that's a negative test result. It wasn't able to grow in that particular environment. I'm just going to go ahead and conclude my video. Thank you so much for watching.